So in this session we are studying T.H.Morgan, the second hybridization experiment. There we are considering the two characters that is present in the same X chromosome, but it is two different characters, that is eye color and the characteristic feature of wing. Okay, here we know the eye color we have already studied. It has a dominant eye color red and it has a recessive eye color that is white. It is represented as, red is represented as W plus and white is represented as W, that we have seen already, right? And drosophila, it will be having normal wing, that is the wild character or dominant character, and the miniature wing or small wing, that is the recessive character. So that is represented as M plus and M. So we are going to take the drosophila as we have done in the previous experiment, the same type of drosophila, like female homozygous recessive and male we are taking the dominant type. So here we will see the female drosophila which we are considering, that has two genes which is representing the eye color and the character of wing. Here the eye color which we are representing, that is white, and the wing character, it is miniature wing. So W and M I am writing here. This is white eye miniature wing, white eye miniature wing. And here we can see the male drosophila which we are taking, that is the dominant variety that has W plus or red eye and normal wing, red eye and normal wing. So here we can see this is representing as M plus, and we know this has single X chromosome and one Y chromosome. During the gamete formation, this will form the gamete or all the egg. It will be containing W, M containing X chromosome. These are the gametes. Okay, the first generation gametes are this, and here it is producing the two types of sperms, one with X chromosome that contain W plus and M plus, which represent the eye color and the wing character, and we get one Y chromosome also in another sperm, right? So when this is joining together, we can get a combination of two X chromosome. One will be having W plus M plus and another one will be having WM, that represent the heterozygous female. This represent the heterozygous female, and here in this case, when this is joining together, we will get the recessive male WM containing X chromosome, which is crossing with one Y chromosome, that gives the recessive male. Now in the next generation, this has to undergo hybridization, right? So here if it simply undergo hybridization, it will form the recessive character and dominant character. It is not going to form any recombinants. You see, this form two different types of gamete, that is W plus M plus containing X chromosome containing egg, and it will be having WM containing X containing egg, and here it will be segregating like this, WM containing X containing sperm and the Y chromosome containing sperm. So here when this is joining with this one, idum kude join cheyda, we will get W plus W, M plus M. This is a female heterozygous dominant variety, that is nothing but red eye normal wing, red eye normal wing. And we can see if this is joining together, we will get W plus M plus male, that show the same characteristic feature as red eye normal wing, red eye normal wing. Okay, now if we see the next one, if this X chromosome joining with this one, it will form WWMM. This is nothing but white eye miniature wing, white eye miniature wing. And next one, if we see WM containing X chromosome, when it is combining with Y chromosome, it will form WM male. This is nothing but the white eye miniature wing containing male drosophila. ഇതിനകത്ത് നമുക്ക് എന്തേ കിട്ടുന്നുള്ളൂ, റെഡ് ഐ നോർമൽ വിങ് ആൻഡ് വൈറ്റ് ഐ മിനിയേച്ചർ വിങ്, ഇങ്ങനെ ഒരു കോമ്പിനേഷനെ കിട്ടുന്നുള്ളൂ അല്ലേ. സോ ഹിയർ വെൻ ഹി ഡിഡ് ദ എക്സ്പെരിമെന്റ്, experimentally when he did all the practical base, what he got the result, you know, he got 37.2% recombinant. He got 37.2% recombinant we got. According to Mendelian genetics, how we got the recombinance in the second generation. It was like 9:3:3:1 was the combination we got in that 9:3:3:1 recombinance in that. This is 3:3:1 phenotype in that 3:3, that is the recombinance, and 9+1, that is the parental combination. So 6 out of 16 we got the recombinance. Appo 16 നമുക്ക് recombinance ആണ് വന്നത്. So here the percentage of recombinance formed in the Mendelian genetics, it was 37.5%. അല്ലേ? പക്ഷെ ഇവിടെ നമ്മൾ ഒരേ ക്രോമസോമിൽ ഇരിക്കുന്നതുകൊണ്ട് what we are expecting, it may get separate during the gamete formation, it may not get separate during the gamete formation, it may undergo recombination, it may not undergo recombination. നമ്മളത് recombination നടക്കുന്നുണ്ടോ ഇല്ലയോ എന്ന് കണ്ടുപിടിക്കാൻ വേണ്ടിയിട്ടാണ് എന്ത് ചെയ്യുന്ന ഈ പ്രാക്ടിക്കൽ ചെയ്യുന്നത്. When we did this practical, what he got, actually the result what he got, that was the recombinance around 37.2%. Recombinant എത്ര കിട്ടി? Recombinance it was 37.2%. ഇവിടെ recombinance എന്ന് പറയുന്നത് എന്താണ്? Red eye miniature wing, white eye normal wing. Red eye miniature wing and white eye normal wing. These are the recombinants here. എങ്ങനെയാണ് റെഡ് ഐ മിനിയേച്ചർ വിങ് വൈറ്റ് ഐ നോർമൽ വിങ് കിട്ടുന്നതെന്ന് നമുക്ക് ഇനി ഇവിടെ നോക്കാം. See here, this experiment in this, when the F2 generations are formed, ഇത് ഞാൻ മായ്ച്ചളയാണ് കേട്ടോ. When the F2 generations are formed, the organism which is included in this cross, that is the heterozygous female, that is a result of F1 cross, and the recessive male, that is also the result of F1 generation, and we are making the cross of this one. This cross ചെയ്യുന്ന സമയത്ത് during the gamete formation inside this female drosophila, or during the egg formation inside this drosophila, what happened here? This will undergo crossing over like this. ഇത് നമുക്ക് crossing over ഇങ്ങനെ നടക്കുന്നത് കാണാം. So when this is undergoing crossing over like this, we can clearly see that this W plus, it will join with M എന്ന് പറഞ്ഞാൽ അത് ഈ arm നേരെ മേലേക്ക് പോകും. അതേപോലെ this arm, upper arm, it will come down, right? So W plus, it will join with M, W, it will join with M plus. So we are going to see how which all ways this gametes can be formed in this drosophila. That gamete which can be produced in this, it will be W plus M plus containing X containing chromosome containing egg. Okay, or X chromosome W plus M plus combination ഉള്ള ഒരെണ്ണം കിട്ടാം. If there is no crossover നടന്നിട്ടില്ലെങ്കിൽ അങ്ങനെ ഉണ്ടാവാം. അതല്ലെങ്കിൽ if there is no crossover, we can see another egg which will be having an X chromosome with both recessive allele. ഇങ്ങനെ ഒരു കോമ്പിനേഷൻ പോസിബിൾ ആണ് അവിടെ recombination നടന്നിട്ടില്ലെങ്കിൽ. If there is a recombination, then we can see this W plus, it will join with the M which has gone down due to the recombination. So here W plus M containing X chromosome containing egg also will be produced. And here we can see the same thing, that is W, it can be joined with M plus, because this M plus, it will come down, right? So W M plus containing X chromosome containing egg also will be produced from here. അപ്പോൾ ഇവിടെ 4 തരത്തിലുള്ള egg produce ചെയ്തിട്ടുണ്ട് അല്ലേ? So here the sperms which is produced from this, it will be having W M containing X chromosome and 1 Y chromosome. ഇവിടെ ഒരുപാട് recombinations in the chance ഒന്നും ഇല്ല. കാരണം എന്താ? Because this is not a homologous pair. The recombination occurs only in between the non-sister chromatids of homologous chromosome. Is it? അപ്പോൾ ഇവിടെ homologous chromosomes അല്ലാത്തത് കൊണ്ട് there is no chance of recombination. We can see this combination. So here this is undergoing cross with this one. When this egg join with this one, I'm not drawing, I'm just writing the composition of those genes W plus W, M plus M. This is showing the characteristic feature as red eye and normal wing, red dye normal wing, and we can see if this is joining with this one, then it produces WWMM, that is nothing but white eye miniature wing. White eye miniature wing. If these two are joining together, then again it will produce W plus W, M plus M. This is again the red dye normal wing, which is showing the female character, is it? Now if we see this is joining with this one, oh sorry, here we can see M plus M malato, M ആണ്. So I forgot this one, I just got confused here. ഇതിൻ്റെ കേസിൽ നോക്കിക്കേ, W plus W M M ആണ് കേട്ടോ. W plus W M M, it gives the red dye miniature wing. Red dye miniature wing, that is a recombinant, is a red dye miniature wing, that is a recombinant. And if we see this joined together, it will form a combination of WW M plus M. It show white eye normal wing. See, white eye normal wing, that is a recombinant. So this is also a recombinant. Now if we see this, the male which is produced from here also, this will be W plus M plus and Y. W plus M plus, it show the male character, it is hemizygous, so we are not writing the alleles in pair. So W plus M plus male, it will be showing red color eye normal wing, and this if it join together, it will form WM male, and this is a white miniature wing carrying or miniature wing containing drosophila. And we can see this W plus M male, it will be having the characteristic feature as red eye miniature wing, and this is a recombinant. And here we can see the WM plus and Y, WM plus, it is a male drosophila, it is white eye normal wing, this is also a recombinant. അപ്പോൾ ഇങ്ങനെ ഒരു പോസിബിലിറ്റി അദ്ദേഹം കണ്ടുപിടിച്ചു, and this was around 37.2%. എന്ന് പറഞ്ഞാൽ 100 ഡ്രോസോഫിലയിലെ എത്ര എണ്ണം ഉണ്ടായിരുന്നു ഇതേപോലത്തെ റീകോമ്പിനൻസ്? 37 എണ്ണം ഉണ്ടായിരുന്നു അല്ലേ? And 37.2, it is not showing much variation from the Mendelian dihybrid cross recombinance ratio, that was 37.5. Now you see the segregation of these two genes, it occur in a frequency which is almost equal to the frequency of the two genes which was present in two homologous chromosomes. Mendelian inheritance നമ്മുടെ characteristic features ഇരുന്നത് ഒന്നാമത്തെ ക്രോമസോമിലും വേറൊന്ന് 7ാമത്തെ ക്രോമസോമിലുമാണ് ഇരുന്നത്. So it can undergo easily separation due to the segregation of homologous chromosome. പക്ഷെ ഇതോ രണ്ടും X ക്രോമസോമിലാണ് ഇരിക്കുന്നത്, but still it has undergone a very good recombination. It has shown a very good recombination frequency, that is around 37.2%. That means these two genes are well separate, and that is enough for recombination during the meiotic division, during the formation of gamete. We have studied in meiotic division, prophase 1 Pachytene stage, recombinase enzyme which will help in recombination in between the two genes. Here if the gap is more, the recombination frequency also it will be more. So here we can say these two genes, it is arranged around 37.2 map units separately, it has been arranged. So the recombination frequency in between these two genes is 37.2% recombination it is showing. So around 1000 drosophila when it is producing, in that 372 number were the recombinants. And in the case of Mendelian inheritance, when we studied these two genes were separated, when these two genes were present in two homologous chromosomes, the recombination frequency it was 37.5. Means out of 1000 new plants which were produced, in that 375 it was showing the non-parental combination. Here 372 it is showing the non-parental combination. So from that it is very clear these two genes, it is loosely arranged. It is not closely linked. So the recombination frequency is more as it is arranged very far. The linkage when it is coming down, recombination frequency it will be going up. So here there are two different types of genes we can see. In the case of same chromosome, there if the gene is arranged very closely, we can say it has tightly linked genes. Linkage is so tight. And if it is arranged a little apart, then we can say that is loosely linked gene. Tightly linked genes and loosely linked gene. Tightly linked gene recombination frequency will be very less, and loosely linked genes, or it is separated genes, we can say the recombination frequency will be high in that. So linkage and crossing over when we say, or linkage and recombination when we say, that is inversely proportional. Linkage and recombination, it is inversely proportional. Okay, so this is all about the linkage and crossing over, and we have to say something, some general points related to this linkage and crossing over. That is, say here, namala linkage parayna samayathu ipo linkage onnupole linkage in which the genes it may be arranged in a cis manner, or it may be arranged in a trans arrangement, we can say. So linked genes it will show cis arrangement and trans arrangement. Adu enganeya namukku nokka. Endana cis arrangement allengi trans arrangement ennokke parayunna endanu namukku nokka. Linked genes it is arranged in the same chromosome, right? So ivide nokkike, if we are saying in the homologous chromosome, A and B genes are present in a single chromosome. If we are considering, if the genes are arranged, both genes, if it is present in, if it is the both genes which is representing that particular character, if it is present in the same chromosome in dominant form, this is called as cis arrangement. Or see, this is the arrangement we call as cis arrangement. ഇനി ഇത് നേരെ തിരിച്ച് അറേഞ്ച് ചെയ്തിരിക്കട്ടെ. അതായത് ഒരു ക്രോമസോമില് ഒരു ജീനിന്റെ ഡൊമിനന്റ് ജീനും അലീലും ഒരു ജീനിന്റെ റിസസീവ് അലീലുമാണ് അറേഞ്ച് ചെയ്തിട്ടുള്ളെങ്കിൽ, that is called as trans arrangement. Cis arrangement of gene in the linked gene and trans arrangement of gene, we can say. And here the complete linkage, where we can see the two genes it will be very closely arranged. Incomplete linkage or loose linkage, it is seen very far arranged gene, we can say. And now there are certain factors which affect the crossing over or recombination. Recombination affect ചെയ്യുന്ന കുറച്ച് ഫാക്ടേഴ്സ് ഉണ്ട്, ഒന്ന് നോട്ട് ചെയ്തോ. ഇത് നമ്മൾ ഒരിക്കൽ ചോദിച്ചൊരു ക്വസ്റ്റ്യൻ ആണ് കേട്ടോ. റീകോമ്പിനേഷൻ ഓർ ക്രോസിംഗ് ഓവർ ഇറ്റ് എഫക്ട് ഡ്യൂ ടു ഏജ്. ഏജ് കൂടുന്തോറും നമുക്ക് ക്രോസിംഗ് ഓവർ ഫ്രീക്വൻസി വിൽ ബി റെഡ്യൂസ്ഡ്. The age when it is increased, the crossing over frequency will be reduced. Age when it is increased, the crossing over frequency will be reduced, or the recombination frequency will be reduced. Temperature when it is increased, the crossover frequency also will be increased. Temperature കൂടുന്തോറും crossover frequency increase ചെയ്യും. Radiation when it is hitted in the gene, this will increase the crossover frequency. Crossover frequency will be, or recombination frequency it will be increased. Okay, so radiation when it is hitting in a chromosome, its crossover frequency will be increased. And the distance between the two genes, if it is more, distance between two genes if it is more, then the crossover frequency also will be increased. Crossover or recombination frequency also will be increased in this, we can say. Okay, അതേപോലെ നമുക്ക് ക്രോമസോം മാപ്പിംഗ് ചെയ്യാം. ക്രോമസോം മാപ്പിംഗ് ചെയ്യുന്നത് എങ്ങനെയാണെന്ന് വെച്ചാൽ, they will give the two different genes are arranged with this much recombination frequency. അങ്ങനെയൊക്കെ പറഞ്ഞിട്ട് recombination frequency തന്നു കഴിഞ്ഞു കഴിഞ്ഞാൽ we can just find it out the gap in between the two genes, and we can arrange according to that recombination frequency we can arrange the genes in the particular order. അതാണ് എന്താണ് ക്രോമസോം മാപ്പിംഗ് എന്ന് പറയുന്നത്. അതായത് ക്രോമസോം മാപ്പിംഗ് ചെയ്യാൻ വേണ്ടിയിട്ട് നമ്മൾ റീകോമ്പിനേഷൻ ഫ്രീക്വൻസി യൂസ് ചെയ്യുന്നുണ്ട് കേട്ടോ. Then the next thing which we have to study, that is some additional topic which they used to ask about crisscross inheritance, non-crisscross inheritance, then we can say like sex-linked inheritance, not sex-linked, sex-limited inheritance, sex-influenced inheritance. ഇതൊക്കെയാണ് നമുക്ക് ഇനി അടുത്ത പഠിക്കാനുള്ള. അതൊരു ചെറിയ ടോപ്പിക്കാണ്, കുറച്ച് അഡീഷണൽ പോയിന്റ്സ് ആണ്. അത് കഴിഞ്ഞിട്ട് നമുക്ക് സെക്സ് ഡിറ്റർമിനേഷനിലേക്ക് എൻ്റർ ചെയ്യാം കേട്ടോ. അപ്പോൾ അത്രയും കൂടെ പറഞ്ഞു കഴിഞ്ഞിട്ടുണ്ടെങ്കിൽ we can complete this portion. Then after that we will enter into sex determination and the next portion. Okay.